Hi class, it's Professor Bittner, and this is the Punnett squares and sexual reproduction segment. I want to emphasize that a Punnett square is just an organized way of thinking about fertilization, but the first step in sexual reproduction is meiosis. So let's say that you have an individual that is big A, little a in their diploid genotype. Then when their cells go through meiosis, about half of those haploid cells, those product cells, will carry the big A allele, and about half will carry the little a allele. When you have an individual that's big A, all of the gametes, all of the haploid cells will carry the big A allele. So the way that we do a Punnett square, I mean, honestly, in this problem here, you don't really need the full grid because one of the individuals has gametes that are all the same genotype, at least for the gene that we're looking at. So remember that the first step in sexual reproduction is meiosis. That makes gametes. In animals, it makes gametes. In plants and fungus, it makes haploid cells called spores, but then eventually the spores make the gametes. It's a little more, a few more steps. But in any case, let's think about the gametes. If the individual is big A, little a, then about half of the gametes will carry a big A allele and about half of the gametes will carry the little a allele. And that's because big A is on one chromosome, maybe a large chromosome, and little a is on the other copy of the large chromosome. And so you remember in meiosis, each of the daughter cells, each of the product cells in meiosis gets one copy of each type of chromosome. So I'm going to get a pen here. Okay, so if you have, for example, big A, little a, it doesn't really matter where you draw it, but draw big A here. And this is the representing the genotype of the gametes. And then if we do a little a down here, there we go. And so that represents, the big A represents that half the gametes would carry the big A allele, or the chromosome that carries the big A allele. The other half of those gametes will carry the little a allele. So that step represents thinking through meiosis. For the other individual, oops, let's do it this way. For the big A parent, all of the gametes, I'm going to try to do it in one motion there, would carry the big A allele. So really, you don't even need this second column over here. You really don't even need this, because all of the gametes from the second parent would be the big A allele genotype. And so recognizing in some cases that you don't need that other column is kind of helpful, I think. And so that's meiosis. Meiosis is figuring out what genotypes the different gametes would be. And that's what you're writing on the outside of the grid. By the way, Mendel didn't use Punnett squares. He just thought about it. The Punnett square came along in the early 1900s with a biologist named Reginald Punnett. And it's just a way of just making sure that you've covered all your bases. But when you, oops, that's wrong. When you bring a big A gamete together with another big A gamete, so the gamete that has the big A up top here fuses with the gamete that has the big A over here on the side, that's fertilization. So meiosis comes first. That's where the gametes are formed. And that's what we write on the outside of the grid. But fertilization is when the gametes fuse together and create a new individual, and that's what we write on the inside of the grid. So these letters I'm typing on the inside of the grid represent the offspring that are possible from these parents. It also indicates what the proportions would be expected to be. What I'm saying is that half the offspring would be expected to be, or there's a 1/2 chance of having a big A offspring from these two parents, and a 1/2 chance of having a big A, little a offspring. But be careful. Only in large data sets would you probably come near to having that kind of ratio. In other words, if 2 individuals of this genotype have 2 children, it does not mean that the one child will definitely be big A, and the other child has to be big A, little a. What it means is that every time the mating happens, you have a 50-50 chance of getting either one of these. Only when the large amount of data is created, in other words, only if they have 1,000 offspring, then you might get data that looks very similar to 50-50, half and half. So each time they have a child, they could have 10 children and all 10 children could come out big A. That's possible. Certainly not the most likely outcome, but it could happen. So only in large sets of data would you expect to get about half of them to be big A, and half to be big A, little a. In small data sets, what you want to do is take these fractions as a proportion or a likelihood for each round of children coming out one way or the other. All right, on number 2, I just gave you something similar. We start with big A, little a for both parents. Let me try it this way. Yeah, maybe this is easier. A big A and a little a, so half the gametes would carry big A, half the gametes would carry little a. And then on this side over here, the same thing. Let me get a pen. A big A and a little a. And so when you fill this in, a 4th chance of having a big A offspring from these parents, a 4th of a big A, little a, another 4th of a big A, little a, and then a 1/4 chance of little a, little a. This is called the monohybrid cross. It's one of Mendels named crosses. So it follows the pattern. I think I mentioned this before. Whoops, I didn't mean to do that. It follows the pattern big A, little a, cross big A, little a as the cross itself, and then the offspring are expected to have, I'll put equals. For phenotypes, the phenotype ratio would be 3 purple, in this case, since big A is purple, for every 1 white flowered offspring. Or you could say 3 out of 4 are purple and 1 out of 4 are white. And this demonstrates Mendels Law of segregation. And segregation is really what's happening in meiosis, that the separation of the alleles happens in anaphase 1 of meiosis, so that if you have a big A on a large chromosome and a little a on the other copy of the large chromosome, that they get separated into different product cells in meiosis. And then the last one here, big A, little a, cross little a, little a. So that one's pretty straightforward. Big A, little a, and then I have to do something else here. Big A, oh no, scratch that. And just kind of pay attention. It's easy to make a mistake, that's for sure, even on these easy ones. Okay, that's supposed to be an A. So, did you need to put the little a twice? No. Again, you could just scratch that because all of the gametes from the second parent would be little a, so you could just scratch that. Make your life easier. And so half of the offspring would be expected to have big A, little a, half would be expected to have little a, but that's only in a large data set. In a small data set, each child born has a half chance of being big A, little a, and each child born has a half chance of being instead little a, little a. So that brings us to our dihybrid cross. In the dihybrid cross, it's the same thing going on. For the first step you do is meiosis. So I'm going to model the meiosis for you so that you can see that. And meiosis tells us what genotype combinations will be in the gametes. And the gametes are what you put across the top of the grid or down the side. And then I'll fill in a few of these, but I'm not going to fill in all of them. And explain how you solve the rest of this. Okay, so let's say that you have an individual who has big A, big B, little b. Now gene A is on one chromosome and gene B is on another chromosome. And so I just found a tool here that I might be able to use here. So let's do the big chromosome. Let me get a pen. I don't know how to get rid of that, but okay, forget about that. Let's do a big chromosome here and another big chromosome here. I'm drawing them in the monad form, it really doesn't matter. And then the other chromosome is the small chromosome. I'm drawing them in the monad form. So starting in meiosis, before prophase 1, you have, well, I can do them dyads, that's fine. But the point is that if an individual is big A, big B, little b, and then what that means is that the big A is on one of the large chromosomes and the little a is on the other. And since sister chromatids are identical, I'm just duplicating that. Of course, there's a replication event prior to prophase 1. On the small chromosome, and it doesn't matter which way you do it, but I'm going to put the big B on this one. Sister chromatid is identical, and then the little b. Now, I could have put the little b on the left and the big B on the right. That doesn't matter. But let's say this is how the individual is. Let's say this is mom's cell, mom. When this cell, and many like it, go through meiosis, what's going to happen is some of the gametes that mom has, eggs, will carry a big A allele and on the other chromosome a big B allele. But it's also possible, because the only rule of meiosis is that each gamete gets one of each type of chromosome, that you could get a big A chromosome and a little b chromosome. And you could also get a big chromosome that has the little a and a little chromosome that has a big B. And you could also get a big chromosome that has a little a and a small chromosome that has a little b. So that's what we're thinking of when we write up here, when we write big A, big B. We're not drawing out the chromosomes, but we're thinking of how meiosis could distribute the chromosomes in different combinations. The only rule is that each gamete has to have one copy of the large chromosome, one copy of the smaller chromosome, and etc. of each type of chromosome. So this would be the 4 possible genotypes. It doesn't really matter what order you put them in, but I would typically put them this way. Now that's mom. Mom makes these eggs. These are her egg types. These are her egg genotypes. Now mom may never have any children. I guess you wouldn't call her mom then. But let's say that woman never has any children. She still makes eggs. Meiosis still happens. So the first step still happens. The man, his body will make sperm. That meiosis will happen. So meiosis makes thousands of eggs and millions of sperm. What's rare is the fertilization, which is the second step of sexual reproduction. Meiosis is common. Sexual reproduction, though, also means that you have to have fertilization, which means that a gamete from who we're going to call dad, because he's going to be a dad in this scenario. Okay, since he's kind of the same, then his sperm would have the same kind of genotypes. And so if mom, the person we're calling mom, and the person we're calling dad conceive a child, if egg and sperm fuse, then you fill those letters in here. Now what's typical is that you put, since I listed A before B, put the A's before the B's when you fill in this grid. It's just visually easier to look at. And when you have, say, a big B and a little b, you put the big B before the little b. All right. So I'll give you an example. This one would be one that would be kind of tricky for students. Put A before B, so put the A's first. And then within the B's, there is a big and then a small, so do it like that. Little a, big B, little b. Do all the A's first, then all the B's, and within one type of letter, do a capital letter in front of the lowercase. Capital letter always indicates that you know that that allele is dominant and will show the dominant phenotype. So then what you have to do is scroll to the questions that are down here, which I can't figure out how to do. Oh, here we go. And you'll have to count through, after you finish the grid, you will have to look through each one of them and decide what color flowers would they have and what color seeds would they have, and maybe put little tabulation marks here. And what you should get is, you should get that there are 9, and it says proportion, so 16 possibilities on the grid. 9 of them should give you a phenotype of purple flower, yellow seeds. 3 of them should give you a phenotype of purple flowers, green seeds. 3 of them should give you a phenotype of white flowers, yellow seeds, and only 1 would give you white flowers and green seeds. And this is the dihybrid cross because the parents, the person we're calling mom and dad, both fit the pattern for heterozygous for both genes that are being considered. And the dihybrid cross always gives a 9-3-3-1, where 9 of them are dominant phenotype for both traits. 3 are dominant phenotype for the first trait, but recessive for the second. This 3 down here is recessive for the first trait, but dominant for the second, so it's kind of the inverse of the previous one. And then only 1 that's recessive for both traits. And that demonstrates what we call Mendels Law of independent assortment.